Hello everyone, I am Nishu and today in my topic I am going to explain you all about incomplete dominance. In my last video I had discussed about your law of dominance, law of segregation, all such topics I have completed in my last two videos. And today I am going to take up the exception of the Mendelian experiment. Incomplete dominance is an exception to Mendel experiment. How? In respect to the result which was obtained. How it is different, I'll be taking up little later. But before starting this topic, incomplete dominance, I would like to explain that what is gene interaction. Gene interaction is one of the most important points which everyone should know, that how and why the genes are being present in the body of any of the living organism. They are interacting. What happens inside the body, that the genes get interacted to each other, that they get communicated to each other. Those concepts are very important to know, though that is a very basic knowledge which is very important for us to understand before understanding all the examples like your incomplete dominance, codominance, multiple allelism. These are all exceptions which was your to the Mendelian's experiment. What Mendel has given an result in F1 generation, they all get deviated from this actual result. So that is why the need of this gene interaction is very important for us to know, that what actually happens, that genes are being getting interacted to each other, how they are getting influenced by each other. And always remember that the genes are made up of alleles, and for every different character there are different type of genes. For the character height there is a separate gene. For the character shape there is a different gene. For the character color there is a separate gene. For each and every character there is separate gene present inside the body. Hair color, eye color, skin color, for everything there is a separate gene which is present inside the living organism, and as they are being expressed, the characters come in front of us as a phenotypic expression. So for that to understand the concept, the real meaning of gene interaction is very important for us to know. So let us know that what is gene interaction. By the term you can understand that gene interaction, the genes are getting interacted to each other, genes are talking to each other, they are communicating to each other, that how to express, when to express, where to express, all such type of question and answers are being going between them, and they are communicating with each other, they are interacting to each other. So this is a language which is not a wordable. We are not verbing it, we are not using any words to express. It is a silent expression which is being done inside the body of any living organism. So what is gene expression? Gene expression can be defined as a type of interaction where the alleles and the non-alleles, if it is an effect, it is a type of, what is gene? It is a type of effect. The interaction is the effect of the alleles and the non-alleles. So how can we define gene interaction? We can define gene interaction as the interaction it is a type of effect. Effect of what? Effect of different type of alleles and non-alleles. That how the expressions, the phenotypic expressions are going to be expressed. So gene interaction is a type of expression, it is a type of interaction between what? Between allele and non-alleles. And where these alleles are present, these alleles are present in the genes, and these alleles are only responsible that which type of character is going to be dominant, which type of character is going to be recessive, and which phenotypic character is going to be observed. And as you know, we have studied a lot before this, that what is allele and non-allele. This concept is not to be make you clear again now, but what is allele? Your allele for a character height, capital T and small t, I am taking it. If I take for a shape, I am taking capital R and small r for round shape. So this is an allele, this is an allele. So allele for character, there are two alleles which are present for a character height, and capital R and small r, these are the alleles which are responsible for the character round shape. So these are alleles of one character and these are alleles of a one character. That means this is a gene and this is a gene. So here what will happen, these are alleles and these two are non-allelic to each other. These are alleles to each other, but these, the alleles of capital T and your small r or small t or small r, these are non-allelic to each other. So how the genes are getting interacted? Genes are interacted in this way. Either the genes are interacted, either the alleles of this character small t is going to act according to the capital T. So either the alleles are communicating here in between them, or the alleles are communicating different to each other in between different two characters. So that is what your gene interaction means. So what does gene interaction means? Gene interaction is the effect of allele and non-allele on normal phenotypic expression of a gene. Hope this is clear. That always, and I told you that always the dominant character will be your expressed in a dominant form and the other of the allele will be following the orders of the dominant one. So here this allele is going to follow the instruction of the capital T, which is dominant one. So interaction is between the two alleles over here, and here your capital T, small t and capital R and small r, these are two different type of genes having two different type of characters. So the interaction can take place in between these two genes also. And what interaction, what is going to take actually interacted? Your alleles are going to interact actually in between them. So these are non-allelic and these are allelic. Hope this concept is clear to you. Now if I move ahead with the same gene interaction to make you more clear this example, that your gene interaction is of two types. Gene interaction can be classified into two types. One, it can be classified as intragenic interaction and the other can be classified as your intergenic interaction. So this is your allelic. Intragenic interaction is called as allelic interaction and intergenic interaction is called as non-allelic interaction. So intragenic is your allelic, interaction, and intergenic is your non-allelic interaction. These are the two types of interactions which is taking place. Now let us come to the terms. What do you mean by intergenic and intragenic? If you remember, I took the word intercellular and intracellular. When I talk about the structures of the parts which are present within the cell, that is your intracellular. But when I talk about the structures of two different cells, that is your intercellular. I hope it is clear that if I am talking, this is one of the cell and this is one of the cell. When I am talking about these two different cells, then it is intercellular. But when I am talking only about this cell and the components which are present only within this cell, then it is intracellular. Similarly, when I am saying the word here intragenic and intergenic, it means that intragenic is this one. I am talking about the two alleles which are present in one gene. So intragenic, it means it is a allelic interaction. And when I am talking about the intergenic, so it means that it is round and it is your height. So I am talking about these two different genes. So this is your intergenic and it can be called as non-allelic interaction. So on this behalf, your gene interaction is classified into intragenic interaction and intergenic interaction. Now again moving ahead, I take an example of a gene situated on a chromosome, of a homozygous type of chromosome or a heterozygous type of chromosome. They are present on a gene on a same chromosome. So if I discuss about intragenic interaction, so two alleles present on a gene of a same chromosome. Always remember, 2 alleles of a gene present on a same chromosome. I am talking about that 2 alleles of a gene present on same chromosome. When they interact between each other, then what will happen? They will produce a product. They will produce something. So they produces phenotype. They will produce a character another character which is expressed, that is your phenotype. So they will produce a phenotype different from normal dominant recessive phenotype. Now what do you mean by this? Now if I come back to the Mendel experiment and I go on this part again, then in Mendel's law you have studied that Mendel performed in F1 generation the first cross, that was he took your dominant tall and recessive dwarf. He cross-pollinated them, and in F1 generation, what did he obtain? He obtained tall plants which was impure, which was hybrid. On cross-pollinating dominant tall and recessive dwarf in F1 generation, he got the result in form of dominant tall, but they were impure, they were hybrid, but the plants were tall. The phenotypic character was tall. But here what the sentence is saying? The sentence says that when the 2 alleles present on the gene of a same chromosome, they interact together, they produces a phenotype dominant tall in F1 generation, which is different from the normal dominant recessive phenotype. Now what was the normal result? Normal result according to Mendel was that the F1 generation plants will be tall, though they were impure, but all the plants were tall. But what is this saying? This is saying that the result in F1 generation will be something else. It will not be following the rules of Mendel's law, first law of dominance. It is not going to follow that rules. So it is saying over here in intragenic interaction. And how it is that? I'll be explaining you just now in this topic, incomplete dominance, that how the results differ from Mendel result and why this is considered as exception to the Mendelian experiment. So here when these 2 alleles present on a gene of a same chromosome is interacting with each other, then they produce some different result in F1 generation. And the examples which we are going to study here is your incomplete dominance, codominance, multiple allelism. And I am going to follow completely all these things from your NCERT book. Line to line I will be explaining it, because these are very important for your board purpose also and for competitive purpose also. A large lot different type of questions are being asked from these topics. So they are very important. Because they being to a exception to the result of Mendelian experiment. So I hope this intragenic interaction is clear to you. Now if I move on to the intergenic interaction. Now what does this mean? This is non-allelic. Now I will be comparing the 2 genes. The genes of your height and the genes of your shape. So when 2 or more genes, now after this I can take your flower color, I can take your position of flower, I can take the stem color, anything I can take over there. So here it is. So 2 or more gene, I have taken the example of 2 gene. So 2 or more gene now present of independent genes. Now they are independent. This is an independent gene responsible for height. This is independent gene for your shape. So 2 or more independent gene present on same or different chromosome. Here it was that they were present on the same chromosome, but here it is not necessary that they might be present on same. They can be present on different chromosomes also. So when 2 or more independent genes present on same or different chromosome, they interact to each other, they produces different expressions, and these expressions are completely different. It is not going to tally here. any of the examples. It is not going to compare with any of the examples of intragenic. The results will be completely different over here from the Mendel's. That is why they are considered to be exceptions. So here are the examples which we are going to study, or which we will be discussing in the later on videos. That is your epistasis, complementary gene, supplementary gene, duplicate gene, lethal gene. There are various such type of examples which can be considered under intergenic interaction. So here this was about your gene interaction, that how your two of the alleles, or your different alleles of or we can say that the two alleles of two different type of genes, how they interact to each other and how the characters are being expressed in front of us. So this is very important to understand, because the terms or the examples which we are going to understand now, or which we are going to study now, so to understand those concepts, this was very important to know before only. So that is why I have explained it here, that what is gene interaction. Then I told you the types of gene interaction. All such things are over here, and I hope this is clear to you. And now I will be moving to the main topic, that is our incomplete dominance. Now moving ahead with the incomplete dominance. Now let us know, by the term only it is clear that the dominance will be there, but it will be incomplete. Now if I take again the example of Mendel's first experiment, in his experiment he took a dominant tall plant and he crossed it with dominant dwarf plant. Now it was a recessive now, and both were homozygous. He crossed between them, and in F1 generation he obtained heterozygous tall plant. Character was dominant. Phenotypic character was dominant. Tall plant was obtained in F1 generation, though it was your hybrid impure type. But in F2 generation, purely the dominant parent character and the dominant recessive character, both of them independently was expressed in F2 generation along with the F1 offspring character also. So in F2 generation, Mendel obtained one dominant tall, one recessive dwarf, and then your two impure hybrid tall. That was the result during the Mendel's experiment. But your incomplete dominance is the experiment which is deviated, which is an exception to the experiment of Mendel. In this type of experiment, now this was performed by a scientist called as Carl Correns, and he observed the result of F1 generation deviated from the Mendel's experiment F1 generation, and it made him think that why such type of results was obtained, and this incomplete dominance, this theory came in front of us as a result of that. So what does this experiment says? This experiment says that now appearance of an intermediate phenotype in a heterozygous obtained when two homozygous parents of different phenotypes are crossed. Now here, which type of parents are being taken? The parents are two homozygous parents having two different phenotypes. Two different phenotypes. It means if I take the character of tall and then your dwarf, one tallness is a phenotype and dwarfness is a phenotype. So homozygous parents are being taken of different phenotypes. Now when these two homozygous parents having different phenotypes are being crossed with each other, then in F1 generation results will be obtained. But that result is an intermediate result. This is what your sentence is saying, that appearance of an intermediate phenotype in a heterozygous obtained when two homozygous parents of different phenotypes are crossed. So in F1 generation, when two homozygous having two different phenotypes, parents are being crossed with each other, then in F1 generation, your heterozygous offsprings are obtained. But the offsprings which are obtained in F1 generation are not similar result to the dominant which was expressed by the Mendel. this is a result here which is obtained is an intermediate result. It is a mixed result of both the type of phenotype. So the appearance of an intermediate phenotype occurs during the F1 generation when your two homozygous parents of different phenotypes are crossed. I'll explain you with this chart over here now. Now, this incomplete dominance is also called as semi-dominance. Semi means intermediate. So that is why it is also called as semi-dominance. Now next point, this is very important over here, that your incomplete dominance do not show blending. Do not show blending because the same parental types are recovered in F2 generation. We will not say that incomplete dominance is showing blending. If it was blending, then in F2 generation, the offspring result would have been differed. But the result is not differing over here in F2 generation. In F1 generation, an intermediate result is being obtained. But in F2 generation, the same parental result will be obtained as it was by the during the Mendel experiment. So that is why here we can say that your incomplete dominance is not showing blending. So here in F2 generation, though in F1 generation, your offsprings are deviated from the original result of Mendel, but in F2 generation, those characters are again recovered as it was in Mendelian experiment. So that is why we will not consider it as a blending of experiment. And I told you just now that this was the experiment which was your first of all observed by Carl Correns, and he has performed his experiment on a very important plant, Mirabilis jalapa. And this Mirabilis jalapa is also called as 4 o'clock plant. And on which character? On flower color. On flower color, Mirabilis jalapa, Carl Correns has performed his experiment. So he has taken the character flower color. Rather than this, another different type of scientists have also worked on this incomplete dominance, and they have worked on Antherinum, which is also called as dog plant or snapdragon, and he has also worked on flower color. And the next example is Andalusian fowl, and on this type the character was taken feather color. And all these examples have been mentioned in your NCERT book, and examples are very important and different questions are being asked on these examples. So all these examples were taken to explain the concept of incomplete dominance. Theory part I have explained. Now moving to the chart part. Now what is it that parents are taken? And I told you, now here I am not taking the character height. I am moving on along with the Carl Correns experiment, that he has taken flower color of Mirabilis jalapa. So here your parent has taken, and this is a flower color. Now which flower color? If I take a capital R and capital R, so it is your red in color. And if I take small r, so it is your white in color. So these are the two traits which was taken by your Carl Correns. Flower color, red flower and white color flower. Red is dominant, having the genotype capital R alleles, and your white is recessive, having alleles small r and small r. And you know how to make gametes here. So your gametes will be prepared, and it will be capital R and small r by crossing both the two types of parent. That is your homozygous. Now what did I say? If you remember the definition, that two homozygous parents are taken of different phenotype. So one is red and another one is white, but both are homozygous. So when two homozygous parent of different phenotype are crossed together, then in F1 generation an intermediate phenotype is observed. According to Mendel, what would be the result here? It would be capital R, capital small r should be your red color flower. But you can see that in F1 generation, pink flower was obtained. So this was a deviated result over here, and it proved the incomplete dominance which was shown by Carl Correns. So here, according to Mendel, this would be red flower. According to law of dominance, Mendel's law. But the results deviated, and in F1 generation, pink flower was obtained. So this was an intermediate result which was obtained. It is pink color is an intermediate color of red and white. So it is an intermediate result which was obtained here, and the result deviated. It is an exception over here. But again, to show that it is not having blending, I told you that here that incomplete dominance do not show blending. How will you prove it? Here, by this first during this F1 generation, you are proving incomplete dominance because the result is deviating from the Mendel result. But to prove that incomplete dominance is not a blending example, so to prove that, what did Carl Correns do? You know, in F2 generation, self-pollination will be done. So from the offspring obtained F1 generation, the two of the offsprings were self-pollinated with each other. The experiment was performed similarly as Mendel has performed. In experiment procedure, there was no difference made. There was no differences. So experiment was performed as such as Mendel, but result was deviating in F1 generation. So moving ahead with the following the Mendel's experiment rules in F2 generation, to obtain F2 generation, your offsprings of F1 generation was self-pollinated. You can consider one as a male and other as a female. So here, making a checkerboard, your Punnett square method, on top I have written male and on bottom side, this side, I have written female. So capital R, small r, capital R, small r. Now making the cross, capital R, here, capital R, small r, then again capital R, small r, small r, small r. So this was the result which was obtained by Mendel also in F2 generation. If you remember, capital T, capital T, capital T, small t, capital T, small t, small t and small t, 1:2:1. Their result was over there. So same result was obtained by Carl Correns in F2 generation by performing experiment on Mirabilis jalapa during your incomplete dominance. So in F2 generation, your result was same, that your 1:2:1 ratio was obtained over here. So your according to genotypic ratio, capital R is 1, capital R, small r is 2 and small r is 1. So according to Mendel's experiment also, what Mendel has performed on the height character, he also gave that in F2 generation the genotypic ratio was 1:2:1. So the same ratio was obtained over here, 1:2:1. So genotypic ratio was same like your Mendel, but phenotypic ratio was changing over here. If you remember, according to Mendel, there the ratio was 3:1. One was pure tall and 2 was hybrid tall and 1 was dwarf. So 3 was tall and 1 was dwarf. But here the ratio is changing. What? Now the results which are changing over here, if you consider capital R, this is your red. Capital R, small r is your pink. Capital R, small r is pink. And small r is white. So what did you obtain? You obtained here one pure red which was from parent, one pure white which was from your parent, and 2 of them were pink intermediate which was of your F1 generation. So what ratio did you obtain? Your phenotypically your 1:2:1. So the phenotypic ratio in the case of incomplete dominance was changing. In Mendel's experiment, during phenotypic ratio, the result obtained was 3:1. But here the results are deviating and you are seeing that the results obtained is 1:2:1. Both the results, your genotypic ratio and phenotypic ratio obtained in case of incomplete dominance is 1:2:1. Why? You can see the result here. According to genes also 1:2:1, and according to phenotype, your character expressed also 1:2:1. So during your incomplete dominance, genotypic and phenotypic ratios are same. Now this type of experiment, this F2 generation is clearly showing, it's clearly depicting that it is not a blending. That during incomplete dominance, the blending of genes do not take place. Blending of alleles do not take place. But what happens in F1 generation, your intermediate result is obtained, while during F2 generation the results get same according to your Mendel rules and Mendel's experiment. So this was all about your incomplete dominance. Now I'll be moving to the next concept, your explanation of your dominance, and that topic is immediately below the topic incomplete dominance in your NCERT book, and that is very important to understand, to know actually that what happens, that these genes are getting interacted to each other. That explanation will be clarifying that how the incomplete dominance or how the traits are being expressed in any of the generation, and in which way they are being expressed. What are the factors behind them which are making them, which are allowing them, the alleles to be expressed in front of each other in any of the generations. So this was all about your incomplete dominance. So hope this is clear to you all, and the explanation part I'll be explaining it in the next video, in the next part. Thank you.